Good morning or afternoon. My name is Emilio Ortiz. I'm here today to talk about a study that we made entitled "The Mitochondrial Genome of Healthy Mice and Humans Contains a High Diversity of Genetic Variants." Mutations in the mitochondrial genome have been linked to aging in humans, primates, and rodents, and cause a range of neuromuscular diseases in humans. When inherited, these de novo or point mutations or indels could cause a wide variety of debilitating and often fatal diseases such as neuromuscular disorders and encephalopathies. Inside the mitochondria, we can find hundreds to thousands of genome copies, many of which are mutation-carrying variants that can coexist with normal genomes in heteroplasmy. It has been proposed that when the frequency of mutations surpasses a critical threshold, it could cause mitochondrial dysfunction. Point mutations and rearrangements have been detected in oocytes, embryos, and adult tissues, which are considered to accumulate, increasing in prevalence during aging and senescence. There is a clear relevance in such mutations for human health. However, little is known about their abundance and diversity in normal tissues. In this work, a high abundance of single nucleotide variants of the mitochondrial genome was observed in mouse and human tissues. Additionally, it was determined in mice that variants arise at high rates in the germline and some that appear in a single generation remain in adult tissues. In this study, the mouse strains CD1 and C57VL were used to obtain brain tissue from adults and embryos, whole liver samples, and oocytes from female mice. Human placenta samples were obtained during programmed cesarean sections from normal-term pregnancies. Afterward, the mitochondrial DNA was extracted from each and sequencing was carried out by Illumina sequencing platforms. Female oocytes were processed to obtain a molecule of circular DNA, followed by paired-end Illumina sequencing. Raw sequences of whole liver and human placenta samples were trimmed using Trim-O-Matic, whereas the rest of the sequences were processed without trimming as they were of good quality as assessed by FastQC. Reads were mapped with SegML to the respective mitochondrial genome reference sequence. The stringent variant calling and filtering were performed with MITEC2. Finally, phylotree was used to identify the geographic or ethnic affiliation of the resulting haplogroups, assessed by haplogrep. To measure the appearance of variants in a single generation in healthy CD1 mice, the mutation load of the liver of 3 female mice and their offspring was analyzed at 3 months of age. The heteroplasmy ranges from 1.1 to 2.6%, and all variants were transitions. These results showed that in a single generation, variants of at least 1% of relative abundance are generated or increased to detectable levels in coding and non-coding regions of the mitochondrial genome in approximately 13% of animals analyzed. To determine whether the variants are generated in the germline or as somatic variants, the mutation load of a triad of oocytes for each of the mouse strains was analyzed. In this table, SM1, 2, and 3 correspond to the C57 strain, and SM4, 5, and 6 correspond to the CD1 strain. All variants were non-synonymous substitutions and were transitions. The BPS rates indicate that the mitochondrial genome variants are generated or that their relative abundance increases during oogenesis. In mouse brain samples, large deletions of diverse sizes were detected and were present in frequencies lower than 0.4%, but which together accounted for a large abundance of variants. The figure shows the total deletion load for young and adult brains and oocytes. For the brain samples, there was a decrease in the deletion load in the mouse strain CD1. In oocytes, de novo deletions appeared in C57, whereas in CD1 didn't. Small abundant deletions were found in 3 of the 8 placenta samples of this study, PC50, 52, and 57. They are located in the energetic region of 2 direct repeats of 9 nucleotides, flanked by 2 important genes. Despite the high or low abundance of each deletion, they share a very similar heteroplasmic profile across the 3 samples. Interestingly, PC54 contains a deletion that coincides with one which was originally identified in East Asian populations and is referred to as the recurrent 9 nucleotide deletion. Since all deletions in this cluster have some degree of overlap with each other, each is expected to be present in different copies of the mitochondrial genome. In conclusion, the increase of age-related variance in rate could lead to their accumulation in specific cell types or structure whilst being undetectable at the organ or tissue level. As observed in the study, it appears to be that most de novo base substitutions are not related directly to oxidative damage to DNA. This strengthens the need to explore new avenues related to the theory that links the generation of variants to oxidative damage to mitochondrial DNA, followed by their accumulation as NG ensues. In half of the human samples analyzed, over 60% of mitochondrial genome copies may bear relations such as group of base substitution of low heteroplasmy in a single individual or those of the intergenic deletions cluster, which likely corresponds to the previously identified 9 nucleotide deletion. And that's it for this presentation. Thank you so much.